right. We're mostly all back, so we're going to get started here. All right. Next on our docket is Kara Snow. Kara is an avian ecologist with Dr. Alexis Grindy's Avian Ecology Lab at the Natural Resources Research Institute, NRRI. She earned her master's from the University of Minnesota Duluth, where she studied the breeding ecology and diet of boreal chickadees. Her work in Minnesota has also included research on tree swallows and golden-winged warblers. Kara is currently contributing to a project documenting nesting habitat preferences of pileated woodpeckers and the wildlife species that use their old cavities. She's also involved in a separate project that's using eDNA found in bird and bat fecal samples to detect invasive insect species in the state. Awesome. Kara.
All right. Well, thank you for that introduction. And thanks everybody for being here. I'm really excited to chat with you all today about some of the neat technologies that we've been using in our lab to learn more about birds and improve conservation. My talk is called Innovation in Action. So I'm going to walk you through some of the tools that we use and how they're helping us answer some of the questions that have previously been really challenged to get at. There we go. So as Clinton mentioned, my name is Kara Snow, and I'm part of the Avian Ecology Lab, which is led by Dr. Alexis Grindy, who's here today. And our lab is part of the Forest and Land Research Group at the Natural Resources Research Institute in Duluth. And we focused on research-based strategies for bird conservation. So basically, turning what we learn in the field and in the lab into real, usable guidance for land managers. And our lab has been doing bird work for over 30 years, so there's a lot of history there and knowledge behind what we do. And right now, we have 7 full-time researchers, 2 grad students, and every year we hire a great crew of field techs to help us gather boatloads of data. Has anybody here worked as a field tech for the Avian Ecology Lab? Oh yeah, I mean, over 30 years, so we've got a lot of folks here. Very neat. Let's see here, where's my little... All right, so why do we study birds? I mean, yes, we generally and genuinely all love birds, but the bigger reason is that birds are really good indicators of environmental change. They react really quickly when habitats shift or when climate or human disturbances ramp up. And as we all know, ecosystems are under a lot of pressure right now. Climate change, habitat loss, fragmentation, the very sad list goes on. And birds are everywhere, and they're in almost every habitat, and they're relatively easy to monitor. So when bird communities start to change, that's kind of like a warning light that ecosystems are changing. Trying to get my little mouse thing to come over. So knowing that birds can act like environmental warning lights is great, but to actually help them, we need kind of a fuller picture of their lives. So we start with some really basic but really important questions, like where are the birds living? What does good habitat look like for them? Where are they going? So Minnesota is often just one stop on a species annual cycle. And also, what are they eating and what affects that availability of food for them? And once we start to get the answers to these questions, then we can start to build smart, targeted conservation action plans. All right, so I'm just going to give us a moment with him for a second. I take a sip of water. That brings us to a bigger kind of guiding question. is the data that we're collecting actually good data? So just because we can collect something doesn't always mean that we should. So some methods are they're time consuming, they're expensive. They can also be pretty uncomfy for birds, right? A lot of the methods that we have involve, capturing or handling birds or entering their sensitive habitat. So we're always asking ourselves, can we get better quality data? Can we get it in a less intrusive way? And honestly, and the point count crew folks who are here kind of understand this one, can we get more data without burning out our crew? So the push to be effective and efficient is always guiding us for looking towards new tools and technologies in our research. All right. So to answer our questions more effectively, we're adding new tools to our toolbox all the time. Some of the more interesting tools that we use are LiDAR. So that's basically super detailed habitat mapping. We have telemetry, so using things like little nano tags that we attach to birds to track them and then to look at the habitat that they use. DNA analysis of droppings, as mentioned, that I do a lot of research with poop. Determining diet through DNA found in poop samples. The use of drones or aerial photography, which is great for remote, documenting birds that are in remote or really sensitive colonies or nesting colonies. And then also using autonomous recording units, which are basically 24/7 bird eavesdropping listening devices. And then also using trail cameras to document wildlife behavior and populations. All right, so LiDAR, I'm not going to go too in depth with this because actually right after me, one of our grad students, Josh, is going to just give everybody a really good description of LiDAR. But LiDAR stands for light detection and ranging, and it's a really non-invasive way to get incredibly 3D detailed info about forest structure. So as you can see with the picture with the plane, an airplane flies over, essentially it shoots lasers down, and those laser pulses bounce off of the landscape, and from that we can get these high-resolution 3D images of the forest. And that gives us structural information that would take a field crew months to collect by hand, and even then, it couldn't compare to the kind of detail that we can get from LiDAR. It lets us see things like accurate canopy height, fine-scale topography, those detailed forest structures, that it can even pick up on cavities in trees. So it's a really neat tool that I'm excited for everybody to learn more about after I talk here from Josh. All right. And understanding habitat isn't just about understanding the forest, it's also about understanding how birds themselves move through the forest. And so for that, we often use tracking technologies. One method that we use involves attaching these tiny, fingernail-sized nano tags to small birds and then tracking them with handheld telemetry. This can give us data on habitat use, post-fledgling survival and movement, and also give us post-fledgling survival estimates. The post-fledgling period is a super vulnerable time for young birds, so if we can see what kind of habitats that they use, we can prioritize conservation efforts in those areas. So kind of to mitigate any population declines that we might be witnessing with those species. Two projects that we have recently used handheld telemetry on are tracking boreal chickadees, learning about their movements after they leave the nest, and what kind of habitat they're selecting. Oftentimes, young birds, juvenile birds, actually have different habitat preferences than adult birds. So really identifying that and learning more about that can be a great insight into their ecology. We've also, we were just learning about the golden winged warbler being a species of special concern. We've used nanotags and telemetry to do a very similar thing, looking at their survival after they leave the nest and what kind of habitats they are choosing. And we also use cellular and GPS tags, which provide real-time location data often without needing to recapture the bird. So again, that's less intrusive. These tags can tell us about habitat use, movement, home range, seasonal behaviors. A project that we're currently working on is putting cellular tags on pileated woodpeckers to understand how different levels of forest management is affecting like their space use and their home range size. And cellular and GPS tags can also help us learn more about migration routes and stopover sites. So these are like really important areas where we can prioritize conservation efforts. So we're looking beyond the state. Many birds, as I mentioned earlier, Minnesota just represents a place where they stay at one piece of their life cycle. And it can also give us insights onto their foraging movements, so where they're eating. That's great, because then we can know where to prioritize habitat conservation or habitat management efforts where they're foraging to increase resource availability. Another really neat and interesting thing that we can learn from these kinds of tags is what individuals are associating with what. So if breeding pairs are traveling together, or if they come back just for the breeding season. And we can also learn about colony connectivity. So are colonies that breed in a certain area, are they all going to the same wintering spots, or do they kind of split up? So again, the idea is like getting the most that we can out of every tag that we put on a bird. Another really powerful tool that we're using is environmental DNA, or eDNA. So this lets us study diet and food web dynamics using DNA found in fecal samples or like cloacal swabs. And it's a way less intrusive method than some of the old school previous methods of learning about nestling behavior, which involved throat ligatures often, or dissection, which is yikes. I think what's really cool about using eDNA as a tool to look at diet is that it really gives us a snapshot of an entire prey community that bird has interacted with, not just what we have seen it eating or what we saw it carrying to the nest. And another thing that I personally get really geeked out about is when I'm analyzing the insect DNA that I've found in a fecal sample, and I learn about a different bug that was found in that. I really like to look up the natural history of the bug, and then I can learn about this bug, it lives on this kind of plant or like this tree species, and then I like to think, oh yeah, what was in that forest? And I kind of like to think of the story of how the bird came to ingest that specific insect. It's just really neat. Diet studies can help us learn about how climate change is impacting prey communities and impacting food resources for birds. And it can also, as I mentioned earlier, give us really detailed insight into insect communities. So it's learning not just about birds, but about that whole other piece of the puzzle. So just like how eDNA expands what we can detect, ARUs, or autonomous recording units, give us another view of bird communities, and they expand what we can detect and when we can detect them. So ARUs can record continuously sounds day or night, and they help us estimate populations and understand community structure. So an example of where we use ARUs in our lab is for our Hunter Walking Trail project, which is looking at how small, frequent harvests affect ruffed grouse and other breeding birds. And we also have used ARUs for boreal owl monitoring. So you can understand how something like this would be good for a species that doesn't really like to let itself be seen, except, of course, like not including last year in Duluth. The place was lousy with them, but ARUs are a device that we use quite often and are really, really important for gathering information on bird communities. But they can also be used for learning more about bats, frogs, children, anything that makes a lot of noise. All right, drones and aerial photography. These are another great low impact, less intrusive tool. And we use these for monitoring remote or sensitive areas where we can't get to on foot, are challenging to get to by boat, and would also just disturb the birds. So we use aerial imagery for monitoring colonial water birds, and we've also used them for checking on the status of breeding common tern colonies. So drones can help us get these accurate counts while, again, keeping disturbance to a minimum. All right, I'm gonna let this play for a second. I'm gonna describe what's going on here first. So trail cameras. The video that's playing now is a camera that was used to document provisioning rates with boreal chickadees. And as you watch, you'll also see that some potential nest predators come in. So they can also be used to kind of identify predators of a species or a nest. We use trail cams quite a lot in our lab, and we use them to document wildlife presence, behavior, responses to management. As I mentioned, they're useful for documenting provisioning rates, and provisioning rates give us an idea about resource availability, which we can use as a proxy for habitat quality. Oh yeah, that squirrel. I like how one of the parents like almost gets it too. The next one's the best though, just you guys wait. So some of the other projects that we use trail cams on right now are our Emerald Ash Borer and Wildlife Project. So that's looking at how different management techniques in black ash forests is affecting like wildlife communities. So we put these different trail cams up in these different experimental harvest techniques just to kind of see what the wildlife communities are looking at in those. That one on the left is a gray fox. We've got a pine marten, bear, magpie. And also the boreal chickadee project. And just this last week, I was up in the Chippewa National Forest with some of my coworkers, very, very cold, putting trail cams up on old pileated woodpecker nesting cavities that we had found this past breeding season so that we can document what other species of wildlife are coming in to use those cavities over the winter and the early spring. All right, well, that was abrupt. Anyways, all right. Well, that's all I have for you today. We have 2 grad students from our lab who are going to talk a little bit more detailed on some of the technologies that they're using for their masters. But with that, I'm happy to answer any questions that anybody might have.
Great talk. I have a question about identifying prey based on eDNA.
Yeah.
I wonder how much genome information is known about various insect species, for example. Can you always identify prey based on the DNA sequence you get?
Yeah, okay, so it depends on a few things. One is, was there enough DNA of that specific prey species for us to amplify? Is that whatever primer or gene that we're using to identify the species? So don't want to get too much in depth with it, but we're looking at a specific gene when we want to identify invertebrates like insects. That gene for that specific species has to have been previously documented and recorded and held in a database. So if that has not been done yet, then we can't, we might have the gene sequence, but if nobody has identified what insect it belongs to, then, we just have a gene sequence. How fresh the fecal sample can make a big difference because DNA starts to degrade rapidly. So if it's a really old fecal sample, then we might lose some of the information that was held in the DNA. So you can get a lot of different bugs, but it's one of those things where it's like, we don't know what we're missing, but it's still a super powerful tool, and we get so many identifications down to the species level. If a fecal sample can have multiple different insect species found in it, it's pretty neat.
Other questions? Okay, thank you, Kara.